Okay, so now we're going to talk about concepts associated with this topic here, postsynaptic potential. And so let me go to a slide that can hopefully start to explain that a little bit more clearly. So again, really familiar slide that we've seen many times. We started with resting potential. We get a stimulus to get a graded potential. If it's strong enough and we reach threshold, we trigger an action potential within the initial segment of the axon that will propagate down the entire length of the axon until we reach the end, and then it is through the synapse and the activity there that we transfer the signal from one neuron to another. Now, it turns out that sometimes we may want to stimulate this postsynaptic neuron, and sometimes we may want to inhibit it. Now, to think of that, I want to go to this view for a minute. Okay, so what I'd like you to think of is you have your hand and you've touched a hot stove. Okay, so we've touched the hot stove and we pull our arm back. Okay, well, we're going to have one set of muscles that we want to contract, these right here. Okay, but these other muscles, we may actually want to relax those muscles. And so we don't want to stimulate. We don't want a stimulating signal here. We want an inhibiting signal. And that then allows us to contract one set of muscles, relax the other, and pull our hand away from a painful stimulus. Okay, so what we've been talking about so far is we've only been talking about stimulating. And to do that, we've been looking at our friend here, the chemical gated sodium channel. But what I want to tell you is that this isn't the only channel that can be on this postsynaptic neuron. We can have other channels that, instead of bringing sodium ion ions, which gets you closer to threshold, we could bring in chloride ions, a negative ion that would bring that postsynaptic cell further away from threshold, so it's less likely to fire an action potential. Okay, so let's talk about each of those types of signals one by one. So first, we've seen this before. Here we're talking about our friend, the chemical gated sodium channel. We start at resting. We get a stimulus, and sodium rushes into the cell, and we get closer to this -60 threshold. If the stimulus goes away before threshold is reached, then that graded potential goes away. In that case, the channel would close, and we go back to rest. So what happens with an inhibiting signal is the opposite. Here, we're at rest, and now we get a signal to inhibit that postsynaptic cell. It brings in negative ions, is one option. And so now you notice that that membrane potential goes actually away from -60. Remember, -60 is up here, so we're moving down away. When that inhibiting signal ends, we would then go back up to resting. A stimulating signal that brings you closer to threshold is called an EPSP. The inhibiting signal is called an IPSP. Notice that you have these vocabulary words. Depolarizing means you're going from resting to above resting, more close to positive. and IPSPs cause hyperpolarizing signals. So here we're going away from threshold, and so we are hyperpolarizing. We're getting lower than resting potential. So in your notes packet, I'd like for you to take that same information that we just went through, and I would like for you to go through this and say an IPSP based on what we just talked about. Is it hyperpolarizing, depolarizing? IPSP, does it cause the membrane potential to get closer to threshold, approach it, or to move away? So go through each of these. One neurotransmitter that can almost always cause an inhibiting signal is called GABA. I'd like for you to know that. One that can almost always cause a stimulating signal is epinephrine. If you'd like a list of other neurotransmitters, here is some of them, where they're found, location, as well as whether they're generally excitatory, inhibitory, both, depending on where they're targeting. Okay. And then here's an activity that will help you go through and continue to learn about the difference between EBSPs and IPSPs. Remember, we're talking about what's happening on this postsynaptic neuron, what's happening to the membrane potential. Notice that potential goes up or down compared to resting. And so that's what this word means, postsynaptic potential.